In this video, we are going to look at where a action potential originates on a muscle fiber, how it moves across the sarcolemma, leading to the contraction of a muscle fiber. So stay tuned. Okay, why don't we get started with this? The image that you see in front of us right now is just a close-up image of a muscle fiber. Okay, so just to orient you a little bit on here, where's my pencil here? There we go. Okay, the pink line that you see going through this way, okay, that's the sarcolemma. That's the muscle fiber membrane. Okay, that's the plasma membrane of the muscle fiber. We've got the myofibrils, which are these long orange color things here. Okay, this is the sarcoplasmic reticulum, all this webbing look. The tubes here, okay, those are the terminal cisternae. The green line going down here, that is your T-tubule or the transverse tubule. Okay. A couple things that we have not discussed previously, which I'll point out briefly here. We will cover it once we are starting the nervous system. But what we're looking at here is this structure. That's the nerve. Okay. This is the end of the nerve, and the end of the nerve, just as an FYI for right now, that's called the synaptic knob, K-N-O-B. So that's the synaptic knob. And where the end of the nerve, the synaptic knob, comes into contact with the muscle fiber, okay, that is the neuromuscular junction. So what we're seeing on this number 1 here, neuromuscular junction, nerve-muscle junction. What we're looking at on here, you can see that the nerve does not come into direct contact with the sarcolemma. Okay, there is a small little space in this area, and that is called the synaptic cleft. And I can't read, these are kind of small here, but that's a synaptic cleft. There's another picture that will get a little bit closer in image of those. Okay, so what we're going to look at is to where the action potential originates, how it propagates or how it moves along the sarcolemma, going down the T-tubule and leading to the contraction of the muscle. So let's look at the motor end plate here, or the neuromuscular junction. Let's look at this one first. Okay, so let's go through some of these. Again, the swollen area here, okay, that is the synaptic knob. Okay, so it's the end of the nerve. Inside the synaptic knob, you see these big round structures. Okay, you've got these big round structures. Those are vesicles. Now, vesicles are just basically a membrane-bound sac. Okay, it's a vessel that carries something. It doesn't do anything, it just carries something from point A to point B. In this case, this vesicle is containing what's called a neurotransmitter. A neurotransmitter is a chemical that goes from a nerve to another structure. So it could be a nerve to another nerve, in this case, nerve to a muscle fiber. Okay, the specific neurotransmitter is called acetylcholine, and it's abbreviated as ACH. Okay, acetylcholine, you're going to hear that quite often in class, and you will definitely have to know what that one is, what its function is, where it's located, all of that stuff. Okay, essentially, how the action potential originates. What happens is there is an impulse, a nerve impulse, that travels down the nerve and it reaches the end of the nerve, the synaptic knob. Okay, so this impulse can, let's say it originates in your brain. My brain says, okay, biceps contract. So an impulse is going from the brain down my neck, down my arm, gets to the biceps, says contract, and the muscle contracts. Let's look at the individual steps at the very beginning of that contraction. And in this case, these vesicles migrate to the membrane at the end of the synaptic knob. Okay, so it's on the side of the synaptic cleft. Think of the vesicle as a juice box. Remember when you're a little kid and you have these juice boxes? Well, the box itself, the container itself, is the vesicle. What that container holds is the neurotransmitter. So the liquid is the neurotransmitter. In the case that we're talking about here, that neurotransmitter is acetylcholine, okay? Now that acetylcholine is released, so if you squeeze your juice box and it kind of comes out, if you release the neurotransmitter, it goes into this synaptic cleft, which is a space between the nerve and the muscle fiber. That neurotransmitter moves across the synaptic cleft, and it moves across by diffusion. If you don't recall what diffusion is, go back and look it up. Look at under passive and active transport mechanisms. So you're going to see simple diffusion, you're going to see facilitated diffusion, osmosis, active transport. Those are the different things that you're going to have a basic understanding of to get here, okay? So simple diffusion in this case is the acetylcholine diffusing across the synaptic cleft. And what we see here, these little gray structures, okay? Those gray structures are a protein structure, and that protein structure creates a channel, okay? If you were to go back and study the components of a cell, we have the plasma membrane, and then that membrane prevents material from the outside of the cell from entering the cell, or from entering the cell to move outside of the cell. That's the function of the plasma membrane or the cell membrane. When we want this action potential to start, we need to change those protein channels. Okay? So what acetylcholine actually does is acetylcholine binds with that protein channel and it opens it up. Normally they're closed, but when you connect it with acetylcholine, they open up. So think of a channel. If they're normally closed, think of it as a your house is locked. Okay, in order to get into the house, you need a key. Well, if you have a key, then you fit it into the lock, then you can open up the door. That's what acetylcholine is essentially doing. Acetylcholine is the key that unlocks the door. When you unlock that door, certain electrolytes can now enter the cell. Now, these protein channels are specific to certain substances. What we're looking at on ours, and those gray ones specifically, are sensitive for sodium. So when we open up that channel, sodium that is located in the synaptic cleft, okay, enters into the cell. They just rush in. And what this is doing is that changes the electrical charge of this membrane, okay? So sodium rushes in here, sodium rushes in here, sodium rushes in here, and this electrical charge builds up and it moves down the membrane. And then it gets to other protein channels. So you can see these are gray. Here we have pink, and then we have blue. Okay, the pink ones are also sodium channels or sodium gates. The difference between the gray one and the pink one or the blue one is that channels can be opened and closed based on the presence or absence of a chemical or the presence and absence of an electrical charge. Okay, so the channels on the motor end plate, which is this region of the sarcolemma, this is the motor end plate, those are chemically gated channels, meaning that channel will open and close based on the presence of a certain chemical. And in this case, that chemical is acetylcholine. Okay, so that's one way of opening up that channel. As this charge moves along, then we get to this channel. Okay, this is no longer a chemically gated channel. This is a voltage gated channel. Now, what that means is that channel will open or close based on an electrical charge. Okay, so as this channel opens up, sodium rushes in, and the blue one is potassium. So the pink is sodium, blue is potassium. So sodium goes into the cell, potassium is going to go out of the cell. When you do that, we're changing the charge, the electrical current, the charge right around those gates. So let's look at another image here. It's just straight down. There we go. Okay, this is the same picture that we had before. We're just zooming in on a different region of it. Okay, so the motor end plate was over here. Now you can see how these channels, these protein channels, are kind of all lined up. Think of those channels or visualize dominoes. So if you knock over the first domino, then knocking over the first one knocks over the second one, which knocks over the third one, and so forth. These channels work the same way. Okay, so as one gate opens up, that changes the electrical charge in that area, and that electrical charge then opens up another gate. When that gate opens up, it changes the charge in that area, which then opens up yet another gate. So here it starts opening up this one, and potassium opens up also. So sodium runs in, potassium goes out. That moves the charge this way, opens up this gate. Sodium goes in, potassium goes out, which opens up this gate. So as you can see the pattern, it's a sequential type of an effect or a domino effect. That charge continues along the sarcolemma. So it's propagating over the sarcolemma. It comes up to the T-tubule. Now remember those little holes that we saw on the sarcolemma, that it's part of the plasma membrane that penetrates deep or invaginates deep into the muscle fiber. So this is still part of the plasma membrane. That charge is still going down the T-tubule, and you can see we still have these channels, so that charge is still going across. Now we get to these channels, okay? Well, as sodium rushes in here, potassium rushes out, that charge opens up this gate. So that's the kind of the beige one here. That's going to open up this one, which is attached to the terminal cisternae. Okay. Now go back and think about a previous video. What is contained within the terminal cisternae? It's a reservoir for calcium ions. Okay. So the charge opens up this one, and there's a few other proteins intermixed in here. What it does, it ultimately releases the stored calcium ions into the sarcoplasm. Now, if you don't recall what a sarcoplasm is, Go back to that previous video. I don't want to give you all the answers here. But it releases the calcium ions into the sarcoplasm. That calcium ions then binds with, kind of hard to see there, troponin. Okay? Now, to relax the muscle, that calcium unattaches or detaches from the troponin, and it's pumped back into the terminal cisternae. Okay? Now, I understand this was very quick, very fast review of this whole process. And again, like I said, this is the sweet spot between anatomy and physiology. Anatomy is the study of structure. Physiology is the study of function. So this is merging structure and a brief introduction to the physiology or the function of it. Okay, we'll go through this more detail. Check out your book. You definitely have to have a sound knowledge on the basics of this in order to move forward, especially when we get into the nervous system. So for right now, we'll call this video complete, and I think I'll start on identifying muscles for lab. See you later.